Hello, my name is Tristan Crawford, and in this video I'm going to be talking about parthenogenesis in humans and how it strays off the path of regular sexual reproduction. I'll also be talking about what would happen if parthenogenesis did not occur and the egg would be fertilized normally. So first off, parthenogenesis basically means virgin birth. Now, this is saying that a female is going to produce an offspring without the aid of a male's sperm. First off, what we're going to have to do is talk about meiosis in humans. Actually, the first parts of human meiosis in a female occur in utero. Human female babies have 500 eggs in each ovary in utero. These eggs are diploid and are called oogonia. It is at this point that the chromosomes separate and replicate themselves. After they have replicated themselves, they join back together, forming sister chromatids. At this point, gene crossover occurs, which creates new sister chromatids, which are genetically different from the original chromosome. The oogonia now splits itself, creating the primary oocyte. Now, after the first stages of meiosis have occurred, they go through a meiotic arrest. This basically means that the process of meiosis halts until puberty. Once puberty is hit in females, they go through ovulation once a month. During ovulation, one of their oocytes goes through meiosis 1. This is when parthenogenesis can happen in females. Basically, what is created is the secondary oocyte and the first polar body. This polar body then disintegrates, so in theory, it's essentially useless. Now, in order for the secondary oocyte to complete meiosis, it requires a calcium spike. This calcium spike is usually provided by the fertilizing sperm cell. Now, this calcium spike is what is needed to initiate meiosis 2. Now, this seems to pose a problem, right? Usually that is the case, but for the purposes of this video, let's just say that that calcium spike does happen. Now, depending on what this person has eaten, there may be an excess amount of calcium in their body. This isn't so far-fetched, believe it or not. Some eggs actually do go through meiosis 2. Since there's a lack of a male sperm cell, the eggs always die. Now, in rare cases, a polar body can actually cause a calcium spike. Once you have this calcium spike, this egg can now go through meiosis 2. When there's no sperm cell present, both of the cells that are created die. Now, in order for parthenogenesis to happen, meiosis 2 needs to not occur. Now, at this point, you're probably thinking meiosis 2 has to occur because the calcium spike is present. Yes, this is usually true, but in some rare cases, there can be a division mistake, causing meiosis 2 to not occur. Now, once this happens, this cell can continue onto starting parthenogenesis. Scarily, when this happens, this egg precursor cell can turn into a tumor. Now, this tumor could be made up of many different tissue types, like liver or teeth or hair, for example. This is true because at this point, all of the cells inside of this precursor cell are stem cells. And as we've learned, stem cells are totipotent, which means that they can turn into any cell type in the human body. Scary stuff, right? At this point, you would think that parthenogenesis has halted. Usually you'd be correct, considering that the unfertilized egg lacks specific instructions about gene expression, which they get from the male sperm cell. In general, our cells have 2 functional copies of each gene, one inherited from the mother and one from the father. For some genes, however, only one copy is ever used, and the other lies dormant. Some of the signals for which copy should be turned off come from the sperm cell. So if there's no sperm, certain genes will be overexpressed, and the embryo will die in about 5 days. I say embryo because at this point it should have been fertilized, but it has gone through specific things in which it hasn't. So in a sense, it's like a false embryo. Although there is a way around this problem as well. In a Japanese lab, scientists were able to eliminate a pair of maternal genes and get a viable baby mouse via parthenogenesis. Surprisingly enough, this baby mouse was unaffected by its lack of paternal imprinting. Even though these changes were genetically engineered, who's to say that they couldn't happen in nature? Because every type of evolution is due to gene mutation. So who's to say that these genes cannot mutate, producing the same results as found in the lab? Obviously, the chances of this are very low, about 1 in a billion. So if we do the math, that's 1 billion times 1 billion. Through that equation, you get a whopping 1 times 10 to the 22nd. If I were to read out that number, it would be 10 sextillion. That's a 1 with 22 zeros after it. Seems like pretty high chances, right? 1 in 10 sextillion. Yes, that would be true, although you haven't taken into account the cell division problem and the calcium spike. Now, I don't have any statistics about the chances of that occurring. So let's just assume it's an astronomical probability. But yes, in theory, it is possible. But for all practicality, the answer is parthenogenesis is not possible. Now, everything that I have said thus far is what would happen during parthenogenesis in humans. Now let's talk a little bit about what would happen if this secondary oocyte was fertilized by a sperm cell. Now at this point, meiosis 1 has already occurred during oogenesis. Now if I haven't said it before, oogenesis is meiosis of female sex cells. So at this point, since fertilization is going to occur, there are several sperm cells that are surrounding the egg. Now you have to keep in mind that during male ejaculation, there is anywhere from 50 to 500 million sperm cells released. So it's only natural to assume that there's several sperm cells surrounding the ovum. Now it's more appropriate to actually say secondary oocyte rather than ovum. So at this point, any sperm cell that is surrounding the egg goes through an acrosomal reaction. It is called an acrosomal reaction because the head of a sperm cell is called the acrosome. Once this reaction occurs, it allows the sperm cell to break through the zona pellucida. The zona pellucida is the jelly coating that surrounds an egg cell, or a secondary oocyte. Once the sperm cell breaks through the zona pellucida, it is transferred into the cytoplasm. At this point, the sperm cell binds with a receptor inside of the egg cell. Now, the first sperm cell to make it through the zona pellucida and into a receptor is the one that is going to fertilize the egg. And once the sperm cell binds with the receptor, the zona pellucida hardens, which prevents any sperm cells to enter the egg. The reason that this is happening is so that the egg prevents polyspermy. Polyspermy is what happens when multiple sperm cells fertilize one egg. Sadly, most embryos die at this point because they are then known as triploids. Triploids have 3 sets of chromosomes, and naturally, when a cell has 3 sets of chromosomes, it will die. So far in this video, we've covered the formation of a zygote through 2 different ways. The first way, via parthenogenesis, and the second way, via fertilization from a sperm cell. Once this zygote is created, it travels down the fallopian tube. This cell goes through mitosis multiple times until it reaches the uterine wall. And with that, my overview of sexual reproduction and parthenogenesis in humans has concluded. More information about these topics can be found here, 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 and here. This link provides information about
fertilization and implantation of the egg in the female human. It's a video which is created by Janix, a company that was formed by the University of Oklahoma, and Next Thought, which is a technology company. It contains some information about how the egg cell is fertilized and its journey down into the uterine wall. It is also where I got some of the pictures that I used to describe sperm cells entering into the secondary oocyte.
And in contrast, this link provides information about
the human parthenogenome. This article is one that is created by McGraw-Hill Higher Education, and it basically goes over all of the reasons why parthenogenesis in humans is simply not possible. It was an article that I read prior to completing the project, just so that I could get a good basis overview of what I was going to tackle.
And finally, in relation to class, we have stem cells.
Obviously, stem cells relate to class because in Chapter 4, we had a whole section about stem cells and their different potency throughout the human body.
And meiosis.
Naturally, since we were discussing the cell life cycle, meiosis has to be included, considering that we went through mitosis briefly, so why not briefly go over meiosis?
So all in all, this concludes my presentation. I hope you enjoyed it. And most importantly, I hope you learned something. Thank you. Once puberty happens, a female goes through ovulation. Female babies have 500 eggs in each uterus. Now this calcium spike is what needs. Usually that is the case, but for the lack of a male sperm cell, they tend to die. No. Yet, but since there's a lack of male genetics, I keep saying that. Now at this point, you're probably thinking meiosis 2 has to occur, since it has gotten the calcium spike, which is what is needed to start meiosis II. That makes no sense. There could be a cell, different types of tissue. And as we have learned, stem cells are pluripotent, totipotent. At this point, you would think that parthenogenesis. So if there's no sperm, now at this point, you're probably thinking that, wait, what? Because of course, every kind of mutation that occurs is due to gene,
Wouldn't the chance be low?
What?
Wouldn't the chance be low?
Yeah, one in 1 billion.
You keep saying high.
What did I say?
So the chances are very high.
Did I say that?
About 1 in a billion.
Did I really say that?
Isn't the chance low?
Did I say high?
You did.
Fuck, how did I word it?
I don't know.
Obviously, the chances of these, obviously, now if you take that into account, that's 1 in a billion for each gene deletion. Fuck, I have to do the math for it now. This topic includes, fuck.